All right, now we are on to my absolutely very favorite biomolecule, and the one that I think is definitely the most useful in biological systems, and that is the proteins. So we're going to be looking at how proteins are formed from amino acids, and a little bit in terms of their complexity of structure, and we will touch on amino acids again when we get into our molecular genetics unit, and we'll be making the connection between how proteins are made in the ribosomes of the cells with how the pattern for those proteins are stored in the DNA in the nucleus. So it's something that we will revisit in Unit 3. So very, very important that you understand all that there is to know about proteins. Okay, as an introduction, there's a few things that are listed here that are sort of ways that we can use proteins in our bodies. This is a very short list, and of course there's a huge long list of what protein actually does in the body. When we think about protein, though, we do think about it as being a key component in cellular growth. So if you're trying to build up muscle and get all buff for the summer, get that six-pack going, then you have to make sure you're eating enough protein. We also are pretty aware that protein is necessary for repairing the cells. And again, if you are working out, then you know that you're developing micro tears in your muscle tissues, and that you need lots of protein to help repair those tears and increase the muscle mass. Proteins are the main components in antibodies that we use for disease. The main component, a huge component of all of the enzymes. And remember, all of the chemical reactions in the body are catalyzed by enzymes. And also in a key component in a lot of hormones, there's a whole part of, or a whole sort of subsection of hormones that are steroid hormones. And of course, those are lipid-based hormones. And when we think of those, we think of things like testosterone and estrogen and progesterone. But a lot of the other hormones and neurotransmitters in our body are protein-based. So we'll be looking at the backbone for proteins, and we may have already discussed that a little bit in class. In the backbone of the molecule, we have carbon, hydrogen, and nitrogen. So the all amino acids have nitrogen, carbon, carbon backbone. We get a little variety in the amino acids on the side chain, which can sometimes contain the phosphorus and the sulfur. Of course, the nitrogen is going to be found on the amino end of the amino acid, and the carbon and oxygen are going to be found on the carboxylic end of the amino acid. So we make these very long chains of amino acids in the ribosome of the cell, and as I said, we'll look at that process in another unit. There's 20 or so. There's a little bit of variation, sort of, again, depending on exactly how you're looking at it, around 20 different kinds of amino acids. Approximately 8 of those are essential, meaning that we cannot make them ourselves, so we have to eat them. And if you're a vegetarian, you probably are aware that you have to work a little harder to match up your food in such a way that you get all those essential amino acids. All the rest of them, the other 12 or so, we are able to manufacture from those 8 that we have. We have different pathways that we can use to develop those. The only difference between all of the amino acids is, in fact, the side chain that dangles off that middle carbon group. And as mentioned earlier, we know that in the backbone of the structure, and that's what this comment is referring to, the backbone of the structure, we have, it should say, at least 2 active functional groups, so please put that in on your note sheet if it doesn't say that already. At least 2 active functional groups are present in the backbone. So we have the amino group and we have the carboxylic acid group, and the side chain may also have some kind of functional group in it. So here's a little graphic of that backbone, the nitrogen in purple and then carbon in gray. We can see the carboxylic acid end with the red oxygen and the amino end with the nitrogen and the hydrogen. The middle carbon is where we find our side chain, and it's just referred to as R here. So we've got those 20 different kinds of side chains that can. So the side chains can be loosely categorized in 4 different categories. They can be polar, meaning hydrophilic. They can be non-polar, meaning hydrophobic. So hydrophilic is water-loving, hydrophobic is water-hating. And if we have a lot of oxygen or nitrogen or phosphorus or sulfur in the side chain, then we're going to have something that's polar. And if we've got just hydrogen or just carbon and hydrogen, we're going to have a side chain that is nonpolar. We can also have basic side chains, which means we're going to have an amino or at least one amino group in the side chain. And again, because it's got some charges related to it, then we've got a side chain that is hydrophilic. And with the acidic, again, we're going to have a carboxylic acid group there, also hydrophilic. So we've got 3 of our 4 groups that are hydrophilic and 1 that's hydrophobic. And that really shouldn't be too surprising given how much water we have in living systems. Now here is a chart of our different amino acids. And in the black lettering, what we're seeing is the side chain. And it's organized a little bit differently than maybe we were drawing it on the board. So if you start moving, pick any one of them, let's say alanine, which is the first one in the upper right-hand corner here. If you look at you'll see H3N, that's at the sort of at the left side of this molecule. So the backbone there is N, which is going to have the 3 hydrogen on it. That means we've also already got an ionization having happened there. C in the middle, and the side chain attached is in the blue. And then we've got C above, which looks like a sort of an upper side chain, if you like, off of the middle carbon. But our backbone is at NC, C above, and that above C, the top C there, is attached to the 2 oxygen, one double bonded to one, single bonded to the other. And the carboxylic acid group has lost its hydrogen as it ionized in water, and that's how they're showing it here. So it's the same NCC background, or backbone rather, that we were talking about, and the side chain is being showed in the bright blue. So if we look at a side chain like alanine, where we've got carbons and hydrogens, we would be pretty quick to guess, I think, that that would be a side chain that would be nonpolar, and it would lend a hydrophobic sort of nature to that amino acid. And valine sitting next to it, again, the side chain is completely composed of carbon and hydrogen, which would be nonpolar. If we have a look at leucine, again, same thing. So all of these ones in the brown region are more or less nonpolar. Now, there could be a little polarity we see tryptophan has a nitrogen down in the side chain, and we see that methionine has a sulfur down in the side chain, but to a large extent, those side chains are composed of hydrogen and carbon. So if we look at the green group here, what we should be seeing is that we've got some if we look proportionally speaking, we've got things like the OH group, so hydroxyl group. We've got sulfhydryl group, the SH. We've got amino groups, and we've got some carbonyl groups. These things are going to add a little bit of polarity. So in the green here, we're seeing mostly polar side chains. Down at the bottom in the pink, you could probably guess what kind of side chains those are. So in that case, we've got carboxylic acid group at the end of the side chain, which lends an acidic nature to that part of the amino acid. So we've got at the bottom aspartic acid and glutamic acid. Those are our 2 acidic side chains. And in the blue at the bottom, we can see each one of these has at least 1 nitrogen group in the side chain, which is going to give these side chains a basic component to them. So these are our basic side chains. All right, so we've talked about amino acids. Now, how do we go from amino acids to actually having a functioning protein, say an enzyme or a hormone or a muscle fiber or something like that? So first step that forms our primary level of organization of a protein is simply condensation reactions that occur between the amino acids. So the amino end of one amino acid is going to bond with the carboxylic acid end of another amino acid. Water will be removed and we'll have our bond. So we can see, well, we maybe can't see so much in our graphic here, but this is the primary structure of protein. And basically, if we start from the left-hand side, we see it's got amino acid number 1. So the N in yellow, NCC, that was the backbone of one amino acid. Then it's been connected from carbon to nitrogen to the next amino acid, which backbone is NCC, and then the third one, and so on. Okay, the bond that's formed there when condensation occurs is called a peptide bond. So remember that, you'll definitely be asked about that. The product that's left, so there's dipeptide, two monomer units put together to form a dipeptide. They still on one end have a carboxylic acid group and on the other end have a amino group. And what you end up with in the backbone is an NCC for amino acid number 1, then an NCC for amino acid number 2, and then an NCC for amino acid number 3, and so on. So in this case, we're looking at the backbone of a tripeptide without having all the other stuff drawn in. And again, here's another diagram of a polypeptide chain in A. And if you have a look, you can see the amino end is to the left and the carboxylic acid end is to the right. And in between that, we have got all of the side chains kind of highlighted in a different color. And we've got the nitrogens and carbons color-coded too. So the blue nitrogen, the middle carbon, which is, I don't know, sort of a black color, I guess, or maybe purple. And then the carboxylic acid carbon, which is in red here. So we can see how we've got these repeating units and we've got the side chains being attached off of the middle carbon group, in this case being drawn down. And have a look at that top bit, because that's going to be coming up and be fairly interesting. We've got hydrogens attached to nitrogen and we've got oxygens attached to carbon here. And we're going to see that something very interesting is going to happen between those as these protein chains, these polypeptide chains are grown. So as I've mentioned earlier, proteins are very complex, and all proteins, well, in order to be a protein, often what we'll do is call them a polypeptide until they become functional. So in order to become a protein, an all grown up protein ready to go, proteins, those polypeptides have to achieve at least 3 or 4 different levels of organization. So these are the 4 levels of protein structure that we're going to be looking at. The primary structure is that simply that peptide linkage that we were looking at a few minutes ago. So amino acid to amino acid to amino acid in a series of condensation reactions. Secondary structure is going to make sort of a little bit more complex, and I like this diagram because it's showing, it sort of looks like a double helix almost. It's not DNA though. We've got this ring, it's called an alpha helix, that's formed, and I'll tell you why as we sort of look ahead a little bit. And then you can see this helix as it's represented inside tertiary structure, where we get something that looks a whole lot more like a blob. And then we get quaternary or quaternary, depending on how you like to say it, quaternary level of structure, where we can see there's in fact 2 polypeptide chains, or actually 4 in this one, 4 polypeptide chains that are working together to do a job. And I'm just looking now at the graphic and seeing that it's got a heme group in the center, which means that this is in fact a hemoglobin molecule, which has 4 subunits or 4 polypeptide chains that work together. So the primary sequence we've already discussed. Again, we're looking at amino acid to amino acid to amino acid in a series of peptide bonds formed through condensation reaction. And it could be any of our 20 in any particular order, however it is that they have been programmed using the blueprint from the DNA in the nucleus and built to order in the ribosome. And again, here we've got peptide bonds being shown in the red here between different amino acids, NCC to NCC to NCC, and so on. Okay, now as I mentioned a minute ago, up in that backbone area, we can see there's an awful lot of potential for hydrogen bonding, or at least I hope that's what you saw, between the hydrogen that we have on the nitrogen group, so on the amino group, and the more polar region of that backbone where the carbon is double bonded to the oxygen group. So we have something that's set up there perfectly for hydrogen bonding. So primary structure involves covalent bonds formed by condensation reactions. Secondary structure of protein is a function of physical bonding, hydrogen bonding, between those parts of the molecule. And depending on what amino acids are being used, so what chains, what side chains are involved, we get a regular pattern of either a coil or a pleated sheet, sort of like a fan that you used to make with folded paper in elementary school. So we can have an alpha helix, or in this note here we call it an alpha coil, but you also see it written as alpha helix, or a beta pleated sheet, and we'll have a look at a diagram of those. So in this little graphic, we've got examples of both, and we can see little dots are being used to illustrate where we're getting hydrogen bonding, again, between the nitrogen-hydrogen from the amino group and the oxygen that's part of the carboxylic acid group in the on the carboxylic end of the amino acid. So we've got hydrogen bonding that forms a very regular pleated, folded structure in a beta pleated sheet. And in the alpha helix, we end up with something that's a little more twisted, but again, a regular structure of interaction, because again, if you look in the backbone, you've got hydrogen, hydrogen, oxygen, hydrogen, hydrogen, oxygen. It's a very regular pattern. Okay, tertiary interaction, the interaction there is a little less regular, because what's happening now is it's not a strictly a condensation reaction that's going to be producing covalent bonds for primary structure. It's not strictly a physical interaction that's going to give us a coil or a pleated sheet. So it's looking at what's happening with the interactions of the side chains. So it depends on what side chains are around, what's going to happen. So kinds of things that might happen are hydrophilic or hydrophobic interactions. So parts of the protein chain could fold away from the water or fold toward the water, depending on which of the two it is. There could be acid and base interactions if we've got the acid and base groups that are that we saw in some of the side chains. We could have some additional hydrogen bonding, not involving the backbone, but involving those side chains, and we could have some ionic or covalent bonds, such as those disulfide links that we were talking about in class. So here's not a horrible diagram of a protein chain. The protein chain here, the NCC is diagrammed just using a light purple line. And we can see here then more clearly maybe some of the interactions. And what we're seeing is up in the sort of left side of this, shown in a light blue, we can see some hydrogen bonding between one side chain and another. In the pink at the top, we can see some hydrophobic interaction. So what's happening is we have sort of a bubble formation in there where the two side chains are kind of hanging out and trying to keep the water out. So it's almost like they would pinch themselves off into a little bubble. In the yellow, we're seeing some disulfide linkages. So we've got a sulfur group from one side chain, sulfur group from another side chain, and they formed a chemical bond in there. So an example of a covalent bond. And then we've got some ionic bonding happening between the acids and bases of two side chains down at the bottom in sort of an aqua color. So some good examples here. And you can see this protein is very irregular in its shape now, and that often happens. In a lot of cases, protein, now not maybe fibrous protein like muscle fiber or microtubules, things like that, that have to have sort of a linear structure to them. But if you're looking at a lot of functional proteins, they're really blobs. They look like a mishmash, a really disarrayed set of chemicals, and it's not. They're very specifically designed that way and meant to take a particular shape based on these interactions. Not all of our proteins are going to reach a quaternary level, so often proteins will be functional at the tertiary level, will not require another polypeptide to work properly. So in some cases, however, this does happen. So we get proteins that require 2 or more polypeptide chains that work together to become functional. You already saw an example of a hemoglobin molecule, which has 4 subunits, 2 alpha and 2 beta chains that come together with a heme group in the center to carry oxygen. Another example that we're going to be looking at in this unit is going to be some very specific kinds of enzymes we call allosteric enzymes, which are the most predominant enzymes in biological systems. And those require 2 polypeptide chains, one that sort of does whatever the enzyme's supposed to do, and then another part that helps regulate whether the enzyme is sort of turned on or turned off in terms of its functionality. There's a graphic of that hemoglobin, and again we can see the heme group and we can see the different polypeptide chains, 2 alpha and 2 beta. So protein are very complex, but there are also a fair few conditions that we can, or that can happen in the environment of the protein that may cause the protein to lose the special shape that makes it so functional, so useful for us. When the protein loses its 3D shape, so whether it's being affected at the quaternary, tertiary, secondary, or primary level, if the shape of the protein is incorrect, if it's been made and then maybe heated or exposed to an acid or put in a very saline environment, that protein will denature. And we do that all the time when we cook our protein. So if we take an egg and we dump it in a pan, it doesn't look the same then as it does when it starts to cook and it turns white. The egg, when it is cooked, is denatured. So that is the term we should be using when we're talking about that. Usually what we're looking for is a pH concentration change, a salt concentration change, or a temperature change. There are lots of other things that will impact on that, but most questions that we'll be dealing with in class or for test purposes will be those sort of 3 things that we'll be looking at that might have caused the protein to become denatured. And this is just a simple graphic showing an active protein and then showing a protein that's been denatured, no longer the right shape to do its job. So there's a little review of the protein structure, and just a reminder that if any of these levels of organization or protein structure are disrupted, then we get a protein that is not functional and won't be able to do the job that we want it to do. It's the end of our protein PowerPoint. So a whole lot more to be said about proteins, but that's all we're going to do here. I want you to think a little bit about other ways that the body might use those proteins, besides the ones that we've talked about here. See what you can find out. Make sure you write down any questions you have for class tomorrow.